So normally somewhere in the rainbow, you should get a folder called something fit. So if you go there, where is it? Where did I put it? So it should be, for me, it will be in my home directory, in my folder. Yeah. So there is several ones. So if you press go several times, you will create, if you go back here, and I'm gonna, yeah, let's just to show you. So in the home directory, I have one here, but the one I just run is this one. If you press several times go, you will put a little plus in front or at the back of the folder saying this is a second folder. And if you press again, you get 2 plus. I'm saying that because I saw someone. So don't worry, take the last one. So take the last one. So now you need to locate where is it. So does everyone has located this folder, which is called yes. So if you go inside this folder, there is a bunch of stuff in it. So the first thing you do, you type Firefox, which is a browser, and report.html. So Firefox report.html inside the folder, okay? In your fit folder. And you press enter. So you should see something like that with different submenu, a little bit like you saw in the fit interface. So let's look at the first. So if you go to registration, it will show what he did, okay? So as I told you, we did several steps. So that is the final step. So fMRI to standard space. So yes. So you have the red lines are the MNI space, and below you have the API images, the functional MRI scan you process. So it's not really well done for the cerebellum, for example, but except that for this subject 5, he did a pretty good job, except the cerebellum. Okay. So you have the different steps. So below you have example. So that is a registration of the API, so the ball image, to the structural one. So the structural is the T1 brain image you enter in the fit menu. And below again you have the API. So you see the affine registration of the T1 to the ball image. Okay. And you have the opposite here. Here you have the structural MRI, and in red you have the API, but it's more difficult to see. And the last line, if you go down, you have here is the registration of the what they call high resolution. This is the T1, your T1 subject to the standard. So below you have the at the back you have the MNI image, the standard space, and the outline. Is it correct? Yeah. And the red outline is your subject file. And the last one is again the functional to the standard. Okay, this is the final step. Yes. Okay, so you need to go inside the folder. Yeah. So the dot fit is a folder, so you need to clean it before to run. Okay. So the reasoning, the red outline is just taking the where the signal is the highest. So it's not very precise. It gives you the outline of the brain. But for example, for the API, you will get a lot of signal from the API outside the brain because it's very noisy. Okay, but doesn't matter. That is just to get a sense of if the registration failed, you will see that the brain is completely upside down. It doesn't match completely the standard space. But here you can see it's matching kind of. And again, so we did just an affine registration. So I didn't go to the nonlinear registration, which is really changing the shape of the brain to match the standard space.
So I said if you did was so you have functional data and we overlap with T1.
Yeah.
And then the T1 will be standard space.
Yes.
Then
And after at the end,
to the
standard space.
okay.
In one go. So you do 2 steps, then at the end,
By the transformation of T1 to standard space, so
Yeah.
then so functional to T1 and then T2.
exactly. Okay. So that is the first step. So normally you should get something. So it's a way to see if the registration is good. If it is not good, there is a way of, again, tweaking the normalization to work. So we change some options to get it sample, but you can change them. So if you go up again, and you go to pre-stats, the second menu. Okay. So you will get something like that. So this is for subject 5. So if you use subject 1, it will be different. So that is a motion correction. So you remember, it's a time series. So the volume, so the subjects can move during the scan. So that gives you the rotation, the translation for the 146 volumes. So you can see that subjects for the rotation, and you can imagine in the scanner, normally we put pads on the side of the brain, on the head. So it's not moving so much like that. So what subject does do more like that. So it would be more on the rotation. So you can see that maybe it's falling asleep and no way. And so that you can see that there is again you can see on. So you have that on the rotation, on the translation. So not so much, but still. So that is for the Z. So you can see that if it's moving, it's maybe moving like that. It will not move like that in the scan. Okay. So that is the relative displacement. So it's a way. So we are not gonna do that, but if there is too much motion, for example, let's say it's too much here, we remove this part and we don't analyze it. Okay, we could do that. Okay. Okay. So normally, and that is stats. It should be empty because we didn't do any stats yet. So let's do it. Okay, so that is the feed. So what else? What is explaining? Let's wait. Let's see. So checking motion. So yes, we did. So again, you can get back to the. So yes, you can. There is a lot of exercise if you want to practice. It's changing bed, changing all the options. Statistics. Let's go. So before, let's look at what we got. So normally you should have FSLI somewhere. Again. So you remember, let's get rid of that. Okay. So if you go to file, add from file, and we go to subject. So if you go to your feed folder, so feed is here. For me, it's here. You have filtered func data. This is your data after preprocessing. Okay. So as I say, we did 10 millimeter smoothing, so you can see it. So let's, I can put more color, but it will not. Okay. So it's very blurry. Okay. So I made that in purpose because I want my signal to be very high, but change it to a little bit lower. Okay.
Isn't it supposed to be a registered?
It's not. So this is in the native space. So the reason for that is we are connecting the statistical first level analysis in the native space, okay? And if we do the second level analysis, you will take the transformation files. So there's a file to move all the subjects in the standard space. So this image is not in the standard space. If you want a glimpse of what is inside the standard space, I can show you where you can find it. You go to File, Add from File. There is a folder in your Fit folder, which is called Reg for registration. So you go inside, and yes, you have all the steps. And normally, if you take yes, this is this one example, func to standard. Yeah, this one. And this one is in a standard space. The only issue is it's only one volume. This example underscore func to standard. Okay. And here, if I go to 000, should be the intérieur comme messieurs. Because, yes. Okay. So it's adjusted to be in the standard space. So I put just 000, which is the origin of the image, and the origin of the image in the standard space should be in the interior commission. You cannot see it on the API image, but from the bold image. But okay. Everything is good. So we did all the registration, all the pre-processing. Okay. So first level analysis. Let's do some statistics. So yes, so it's a time series. Okay, so we got it. So that I don't need. So I will not go over the bold signal. So this is what we got as signal. So it's a bold blood oxygenated level dependent signal. So it's using the signal from the deoxygenated bloods. So there is some explanation. So this is the signal we get. So it's not a direct measure of the neuronal activity. It's a measure of consumption of oxygen, which give you a proxy for activity of the brain. Okay, but it's an indirect measure. Let's keep that in mind. Okay, so that it's right in here. It's an indirect measure of neural firing. But what that means, it's also that when you deal with this kind of signal, you need to keep in mind there is a hemodynamic response. Okay, so it's not on off that is happening. So when you have a stimuli, for example, you need to model the signal to be following HRF, so an hemodynamic response function. So let's just show you briefly what is it. So here's a really nice animation. So you have a stimuli. What is happening for the signal? Just after the stimuli, you have this increase of signal and decrease of signal. So it's delayed in time. So it takes some time to get the oxygen and to get rid of the oxygen. So if you have several stimuli, you will get signal like that. And so you can imagine if you have stimuli that are following each other, so first stimuli, a second one, but it's superposing to the first one and the third one, okay? So when you do statistics, you need to take into account this modelization, okay? So the signal will move like that, okay? Okay, so yes, and if you go back to, there is different example and all the information you can get here about the HRF. But so two things. So first of all, it's an indirect measure, the bold signal, and you need to take into account the hemodynamic response function. Okay. So now let's get to the point. So what you want to do is statistics. So remember, so what we want to correlate is the effect of the task, which is, go back to the frontal task, we have congruent and incongruent stimuli. So what we want to do is to see, for example, if there is a difference of activity between these two conditions, okay? So what we can do is to use general linear model. So who is familiar with statistics and general linear model? Okay, okay, so I don't. Just to make it simple, so when you do a t-test, so t-test is okay for everybody, ANOVA, okay? So when you do that, just a subtype of general linear model. So because the general linear model is just using this kind of equation to model your data. So just, yeah, you see y is your dependent variable and x is your independent variable. Let's, I don't know, you take IQ here and age here, and you want to model IQ based on age, for example. Okay? So that is your dependent variable, and you want to explain it, for example, with age. So for each parameter, for x1, which will be age, you want to estimate the beta value, which is the beta weight, the parameter estimates. Okay. So that is mainly that. So you try to, so you have this data, which is IQ, you have this data, and you have unknown variable that you want to derive from the model. And as life, everything is not perfect, you will get some residual. So you cannot match completely the both to be too perfect to match or to find a correlation between age and IQ to be completely correlated. Okay? So this is what we will do. So we try to model to try to so which way that would be function yeah, that will be your data, so the flanker task, and that will be the bold signal. Okay, and we will do that for every voxel. Okay, it's a time series, so here you will get the bold during the 146 volts for one voxel. And here you will get the flanker task function here. Okay? Is it?
Okay, so blanket tasks, two conditions, congruence for.
Yeah, so yes. Let's see how we model that. So that is okay. So yeah, I will not go into detail. Yeah, that is an example. Yeah, that is a IQ. Yeah, okay. That was IQ and drinks per week. It was not age, but you can do the same thing. Okay. Okay, so if you go back to the folder where is your data, let's go back here. Yes, that you can close the report session. You don't need it anymore. So if you go to one subject, any of them, or the one you just processed, so I go back to work. Yes. So sub, let's go to sub 05 for me. And if you go to functional, don't look at congruent and incongruent text file. Normally, you should not have them. Okay. What you have normally is a TSV file. Okay. Events.tsv file. So it's tabulated space value. Okay. And if I do, just to look at inside the file, I do more. You go inside, so I repeat, you go inside the folder of your subjects in front. There is two files for the, so you have the bold image, and below you have the TSV file for the session run one, and for session 2. Okay, and so if you do more, and yeah, just to look inside the TXT file, it gives you that. Okay. So if I remember, yeah, okay, it's pretty simple. So you have the onset of the stimuli, the duration. So it matches the time of the TR. It's incongruent, correct? And you have the onset and the duration of all the stimuli you have during the scan. So you have incongruent, correct. You have the response time, yeah. You have the if it is correct or incorrect, if the subjects did a mistake, okay. And let's forget about the rest, okay. Make sense?
In real life, where does this data come from?
From the scanner.
For the convert patients, which is correct, which is not.
So normally you have so practically, you mean on the scan, you use a computer that is displaying something on the screen. So you can use what the name of the yeah, you can use that. Also another one, which is I know. Yes, ePrime is a software that record all the response of your subjects, and after, yeah. Yes, that is not new imaging at all, just behavioral data. Okay. So ePrime is yeah, it's the one in Python is yeah, it's free, and it's more complicated.
But they have the filter, which is
Pretty simple.
quite intuitive. They have tutorials online.
Okay, so no, yeah,
They have like a filter interface, and with the tutorials online, it's quite easy.
okay, so it's better. Okay, so
Yeah.
I don't know this one.
Sorry. Open Sesame. So Open Sesame runs Cyclify, but you can run everything on.
Okay, I know. Okay, good to know. Okay, so yeah, again, this is just behavioral data based on your. So what you want to correlate, as I would say, yeah, this is the type of stimuli the subject is doing. Okay. So FSL will not accept this kind of file. So there is a trick again. If you go back to the GitHub, yeah, we need to format the response. So you need to do exactly that. So that, let me put it a little bit. Everybody's seeing it. So you need to copy and paste this. So you go to the dataset folder. Okay, and you run this first command, which is downloading a small script to convert the behavioral data to something that HSL can read. And you run it using a bash script. And it should do that automatically. So let's try to do that. On terminal. So you need to go to a terminal, a new terminal if you want. You go to the folder of your data set, doing CD yes 000102. Is going well? Okay. Let's check. So I had a question saying, but why are we doing that? Yes, it's the right question. So FSL will accept a certain kind of TXT files. So the function you just ran is just formatting the behavioral data to be fed in the FSL in the fit statistical module. Okay, just doing that. So if you go back to this folder, you should see. So this is the original file, the board and the behavioral data, and you should get these 4 supplementary files. So you have congruent run 1, congruent run 2, incongruent run 1 and incongruent run 2. These files are in the func folder. Okay. Everybody has these files? Check, just check. Everybody?
Yeah.
Everybody? No issue. Okay. Very good. So let's look, for example, we'll use the session 1. So we will use the run 1. So let's check at the first file. Okay, more. So what you see is just a file, very short file, with, for the congruent condition, the onset of the stimuli, the duration, and yeah, the condition one. Okay, and which we do the same thing for incongruent. One. You will get something similar, except it's for the incongruent stimuli. You have the onset of the stimuli, the stimulus, and the duration and the condition, which is also one for this condition. Okay, just 3 columns. So just reformatting the text file, the one you just saw before, to have the incongruent and the congruent in 2 files. Okay. No, now what we want to do is to correlate the both signal with this stimuli. Okay. So the second step will be to put the HRF, the hemodynamic response in it. So it will do automatically that. So it will take the location of your stimuli. It will modulate your signal for you will model the signal based on the HRF and try to correlate that with your whole image. Is it clear? So you will do just a correlation between that and the whole, modulated by the HRF. Over to the signal you want to get.
So yeah, which one of the TXT5 responds to what?
A stimuli.
No, here it's the timing.
Yeah, the timing, the onset.
And the second is
Duration.
the duration.
Okay, so it's yeah, the first stimuli is incongruent.
Yeah.
And it's 2 seconds, and yes.
Okay.
Okay. And you have that for all the stimuli.
Yeah. Thank you.
You good? Yes. Even I can show you now. So let's see at what point here we are. If I go back here, if I'm missing. So the general linear model. So yes. So Y will be the ordinal and X will be the congruent or the incongruent, both of them. So just to come back to this equation, let's forget about the HRF, but what we want to do is to do a correlation between the ball, congruent, and incongruent. And we want to estimate this parameter here to see if it is correlated. Okay. So the parameter estimate will give you a number, and you can do statistical. You will get the p-value for this parameter, giving you if it is correlated or not. And so it's every voxel. For every voxel, you will get a number saying if this voxel is correlated to a congruent stimuli or an incongruent stimuli. Clear?
Mm-hmm.
Oh, yes. So that is the part. Time series. Okay, so that's just to explain you how you fit. Yes. So that is your board data, and what you want to do is you have a stimuli, you modulate by the HRF, and you see how much your signal is matching. If it is matching completely, the B value, the parameter you get will be high, and the P value too. Okay. So you will try to do that for all the parameter, creating, yeah. So it's talking about the HRF. So that we did that. So what we did just with the script was taking this file and converting it to something like that. Okay.
We're separating models So one model for congruent trials, another different model for incongruent.
No, it will be, so it's why I show you the general linear model. So we will put everything in the same equation. So you will get one parameter for congruent, one parameter for incongruent. This is a parameter estimate. You will get one image per parameter. Yes.
Because it's just like a general association of the load response with like any.
So if you want to take into account the response, you need to add another parameter in it. And you can take care of that.
But it shouldn't that be like or.
Yeah. Yeah, for the tutorial, I will not go over that one, but yes, yeah. But for sure, you would not get the same signal for incorrect or correct
Yeah.
answers. Yes.
Okay.
The corresponding.
Yeah.
Yeah, the corresponding. This is just taking the correct. Okay,
Yeah.
so it's removing the incorrect response. Okay, so this way you don't care about the correct. Okay. Thank
I mean.
you. So this is very simple. Yeah, they don't care about the incorrect. They want just the correct response for congruent and incongruent. Okay. So let's do it. Finally. So you go back here. So yes, let's close it. Let's go back to so what you can do is to reopen Fit. So the best is maybe to close it. So let's find it back. Where was it? Not here. Not here. Did I close it? Okay, let's. Yeah. So you open. So close your Fit folder. Your Fit module. So this one, you close it and you reopen it. So we will start. In a way, I can show you something. So you get a new one. Okay, so we start back to empty folder. Okay, empty menu. Okay. So you close it, you reopen it. Okay.
But so need some time to start.
No, not after. Yeah, you sure? So if you go here, for me, yeah, the first time it takes time, but after. So don't close it for you. So what you do, except you do statistics, okay? So here you can select as an input, you click here. Input is a fit directory. So input will be the fit directory we just generated for the preprocessing. Okay, so you click on it and you need to select the fit directory. So you click here, there is a menu. You click on this small folder and you go looking for your fit folder, which is somewhere. So for some person, it's inside the func folder. For other people, it's in the home directory, and for other people, it's lost. So everybody knows where is the fit folder. Okay. So you select it. So mine is here, and okay. So here again, when you click, okay. What? Which error? Ah.
Down there. Down there.
What?
Why I am that?
It's down on the left.
Ah, yes, shit. Warning. Ah, yes. You have a warning. Don't worry about the warning. Okay. Okay. All right. So you click the warning is not important. You click OK. And you should get again to check if it is correct. You should see that it's 146 volume. Here. So now, issue. So we loaded our fMRI data, so that we don't care. So let's go to stat. So we'll do a full model setup. So in the stat
stats tabulation, you have a full model setup. You click on it. Okay. You get that, okay? You have this kind of menu. So you have something called EV, which is explanatory variable. So you remember the general linear model? You have y, x. So that is x, okay? Remember, we have two x, because we have congruent and congruent. So number of original EVs will be 2. Okay, so now you have two submenus, 1 and 2. Clear? So the first one, let's say it's congruent. So I will call it cong for sure. Okay? And after you select, so here is asking which kind of data you want to put in. So we will use the custom 3 columns format. So the 3 columns are the one I just showed you before, the onset, duration, and the condition. So you type here. Okay? And you go look for this file, which is the congruent. So you go back to the folder, subject 5 or 1 for those who are using. So let's take only the run, the first run 1. So you take the congruent run 1. Okay. And you do the same thing for the second EV. So let's call it incongruent. Again, custom 3 column format. And you look for this file. Ah, shit. 5, 5, and incongruent. Run 1. We're good. So let's remove temporal derivative. It's for this example, we will not use it. It's a way of taking so you add a delay or a shift in your HRF to try to model if there is a delay in your bold response. But for the tutorial, we will not care about that. So let's keep it simple. So you remove this add temporal derivative, okay? And for both, for EV1 and EV2, for congruent and non-congruent. So you unclick this temporal derivative. Okay. So that is the equation. Okay. So you will you have the convolution by the HRF, which is a gamma thing with different parameter. We will not touch it. Okay. So it's doing the convolution of the HRF for you for EV1 and EV2. Remember the equation is y equal x beta 1 plus x beta 2. What you want to get is the parameter, the beta, the parameter estimates. So when you get them, you want to do statistics on them. So there is a second menu, contrast and F-test. You go there. So it's where you specify which parameter you want to test. Okay. So I will do three contrasts. So I put 3, 1, 2, 3 here. Okay. So you have 3 lines for each contrast you want. And so the first one will be congruent, the congruent parameter. So that each column, the first column is EV1, the second column is EV2. If you remember, EV1 is congruent and EV2 is the incongruent. So if I want to test the parameter estimate of congruent, I put 1 here in the first EV. Okay. And the second one will be incongruent. And that will be 1 in the second column. And the last, what can I do? Huh? Yeah. So let's do incongruent versus congruent. So I ask you which one is the most difficult. So what we want to see is for the incongruent, where you need to do some inhibition. What are the regions that are more activated for the incongruent stimuli compared to the congruent? So we do 1 and -1 minus. Is it minus? No, not.
Just vector forward.
Yeah.
Then just congruent and then.
The difference between the two. So in fact, it's just you take parameter 1 versus a negative parameter 2, and you see if there is a difference between the two parameters.
That would be the slope of this individual.
Yeah.
That would be the slope.
The correlations between the, yeah. So let's check. Normally we are set. So normally if you done, so you will get something like that. Can I open it a little bit bigger? Is it possible? No, it will not be bigger. Okay, so just yeah, it's a summary of what you did. So that is what you observe. So this is congruent and incongruent. What you see is this is convoluted by the HRF. Okay, it's why you see an increase, a decrease. So try to model the perfect signal modeling based on your behavioral data. So you should see something like that. If the brain was perfect or the life is perfect, you should follow this kind of thing for congruent and incongruent. So you will try to correlate this signal with the both signal. And you will find every voxel that is statistically significantly correlated with that. Okay.
So that's the experiment on the front.
Yeah.
Okay.
Are you familiar with this game? Maybe more? No.
Yeah.
Okay. So it's something similar, but it doesn't manage it. It doesn't do the same thing. And here is the different contrast. You set it up. So 1 for congruent, 1 for incongruent, and incongruent versus congruent. Okay. Okay, so normally, okay, so we did that. So I will go just brief. Okay, so just to answer the question, if you want to automate, so what you can do is to save. There is a button here to save this script. So it will save a txt file. So that was the question. So if you click here, it will save inside the fit folder. You will see a design dot FSL or FSS, I think. And everything is saving it. And so if you do fit, not capital fit, but fit, normal fit with the name of this file, you rerun it. So you can inside just a txt file with the name of this folder, what you're doing, the pre-registration, the registration, the smoothing, everything. So you can change the FSL file to do what you want. So if you want to do more subjects, you just need to do a loop running through this design .fsl file, and you can run it for several subjects at the same time. Yeah. So if you are familiar with it, yeah, you can use either bash for each folder, do that, and it will do it. Last menu, but I will not change it. So that is a statistical part. It's where in SPM you can do that after, but here you need to specify which statistical threshold you want to use. So you can use cluster-based statistics. So you set the p-value here, or you can select voxel, corrected voxel threshold. So it's taking care of the multiple comparison problem. Okay. And the last one is uncorrected. So when you are exploring your data, you don't care so much about the correction, and multiple you can do uncorrected, and you put the p-value here. So, but let's go back to cluster. I think, oh yeah, thank you. Okay. So I think we are set. Can we run it? Is it possible? Yeah, let's try it. I think everything is set. So when you are ready, you click on go. Mhm. No, it will run inside the feed directory you entered. So you don't need to change it. So normally, so yes, and you should see it here when finished. So a way to see if it is progressing. If you go back to the feed folder, where is it? Sub. If you go to the feed folder, you do Firefox report again. So you track, yes, not now. Okay, so you remember you had the registration tabulation, the pre-stat. Now the stat, you have something in it. So you have a recap. Again, the design, your matrix design, the contrast. Okay, and post stat. Is it finished? Yes, it's finished. So it gives you, it's a summary of your results. The first parameter estimate, which is with a threshold, okay? The second parameter estimate, and the last one, which is the difference between incongruent and congruent, okay? Yeah. So nothing is happening in which subject?
Subject one.
Subject one.
Yes.
I don't have anything.
I was lucky. I took. So this is the limitation. So we are on individual level, but it's a first level. So you can get signal, for example, for this parameter, we look at it. But looking at that, you need to go to a group level to get something from it. Because yeah, if it was too easy, it's only one subject to get.
You have to do this.
So that is the again okay, so that is interesting. That is a way of showing how your data are fitting. So the cup is your so in red you have your data. And in purple, in purple, how your data is fitting, how you fit your model to the data. So if it is perfect, we'll have a complete overlap between these two lines. So if we, go. So now, just to show you the last thing, if you go back to the FSLIs, you can go back to the functional folder. Yes, it's here. So that is the threshold stats, the one you just saw. So this is for the first contrast. So threshold, this stat 1, this stat 2, and this. So let's take the first one, for example. So it's threshold, this stat 1. Okay, there is nothing, but yeah, because I'm not in the proper folder. Let's remove the. Yes, so let's. What we can do is to add the. Let's take the. So let's take the filter func data first. Okay. So there is some little arrows here where you can put the one below or above. So I will put my API below and my Z-stat above. Okay, like that. Okay. And let's change. So you click on the Z-stat, you select it, you change it to this, like that. Yes. Okay. And that is your Z map. So this is the statistical map for the first parameter estimate, the first contrast, which is the congruent condition. Okay. And you can add, yeah.
If they want to report them in the paper,
Yeah.
how do they export these coordinates or like?
So either they,
X, Y, Z and the cluster size and the Z value.
yeah, there is a command line you can use, which is report statistics FSL. So you give the image and what is the name? Yes, okay. Let's. So one last thing. So you can do that for the different Z score. And something you can do is also to go to stats. Now you have a folder called stats in the fit folder. And if you want to see the unthresholded maps, they are here. So for example, for the first contrast, we have the Z stats one, which is not thresholded. So you can open it. So this is the D value for all the voxel. Let's put some color in it. Let's put, you have the minus. So let's take render 3 here. So you have the positive correlation in red and the negative correlation with the congruent condition in blue. Yes. For subject one?
Yeah.
Yeah, it was, I was saying, so it's a first level analysis only one subject. So you need to go to the group level to get something now. So what you can do is to go to the, so you select the Z stat 3 in the, if you want to see some unthresholded results, you go back here. So this is unthresholded, but you can see where it should be positively correlated and negatively correlated with in the congruent, incongruent minus congruent condition.
And in the negative correlation, like you select all the cases, or you have to, there's more people to do analysis.
Yeah, you need more people. You need to have. Yes, that's why I told you you can save the design.
Yes, that's.
So it's yeah. And after you do loop to run it through, it's why I didn't want to do it here because it would take time. But yeah. Yeah. So just to wrap up because it's already yeah. So we did that. So this is the original paper. It was in 2001. So this is the paper that is related to the data set you are using. So this is the first contrast. So this is congruent, I think. Congruent and incongruent Z maps. Okay.
It's for all the subjects.
For all the subjects. Okay. But if you look at your data, you will find some aspect of it. So you see that, for example, so they don't look, so deactivation is in purple, and they look at activation. So you see that the activation here is a little bit higher here than here, and for other regions too. So medial, prefrontal, and stuff like that. So if you go here and you look at the congruent, so which is this one, and we look at the So I think subject 5 is not so good, but you can see, for example, here, you can see the spot here that the one we just show in the congruent condition. And so you can find at the individual level, even some patterns that are similar to the group level. So what we didn't do is, so you can compare the data later on based on this paper, Kelly et al. 2001. And so recently, what we didn't do is that. So what we did was that. So we have a time series. We ran the first level analysis. We got this parametric map at the individual level. So what you do, you go back, you do that for all the subjects. You get all these parametric maps, and you go to a second level. And so, for example, for incongruent versus congruent, you do that for all the subjects. You get the proper.
To do this, you have to normalize all the subjects in standard space.
Yeah, but it's what we did. Not to the standard base. You don't need to. It's an overlap. It's a statistical map at the second level. So it's not overlapping, but you will test each subject will be a data point. So what I want to here, you do a correlation between your stimuli and your both image. Here, it's just one image per contrast. So it's one image per subject. So you take all the subjects, there are 26 subjects, and you do a contrast between for the 26 congruent image versus, or the opposite, the 26 incongruent parameter estimate versus the 26 congruent parameter estimate images. And you do it's a t-test, in fact. Okay.
Struggling with the number of trials for congruence versus incongruence is different.
It's unbalanced. It's not good to have this kind of condition because the correlation can be higher in one condition than the sampling of your data, of your time series will be different. So it's not ideal, but it's better to have balance. In any kind of statistics, it's better when you are comparing the same. So it's like if you are comparing 200 subjects versus 10. It's not really a good way to do it. And yes, so normally when you do the group analysis, you get these very nice images where you have this medial and lateral prefrontal activation in the condition incongruent correct versus congruent correct images. But that is for another time. I'm sorry, I wanted to do, but that you can do that by yourself. The functional tone. I mean, if you agree, we can just look at a glimpse of it, or we can stop here. So as you want. Let's continue. Yeah, always. So we finished with the first level analysis and stuff like that. Let's see what we want to know, what we want to do now, right? Is to do functional count. So Michelle explained already what we are doing. So we have this time series we got now because we did the first level analysis. So we have different time series for different voxels, and what we want to do is to project this signal in the Y matter. And it's a way of, so these voxels are, all these voxels are based on the Y matter using priors, are connected. So we weight the signal from these three voxels to, not doing a mean, but weighted the signal from these three voxels to get the signal, to get the new signal from the white matter in this location. But we shall already make the explanation of it, but just to, so there is a command line we will use here. It's on the GitHub again. So if we go there, so let's take the first command. You do that, so yes. Okay, so let's close everything. Let's get rid of everything. Okay. Let's open a new shell this time. So we'll install Functionecton on the Neurodesk container. I open a new Is it working? Let's close it. FSL. Bye-bye. So I want a new shell. Yes, new shell. Oh, let's go to zoom in. Zoom in. Zoom in. Zoom in. Is it big enough? So you take the first command line in the GitHub. So if I go back to the GitHub, okay, so that we forget about spatial normalization, so we don't care. So that is pip install that. So you copy, you go to the Nodesk, in your new shell, you do paste as you can. And normally at the end, you have successfully installed function ectone.